Hello, my name is Daniel Richardson. I'm a consultant in sexual health and HIV medicine in Brighton in the UK. I'm one of the editors of this e-learning for health sexually transmitted enteric infections learning tool. And as part of this tool, we produce 3 short videos to illustrate and highlight some specific areas around this topic. This video is around the history and some of the current issues in sexually transmitted enteric infections, specifically antimicrobial resistance. And I'm going to talk mainly around Entamoeba histolytica and Giardia, which are both protozoal infections, hepatitis A and Shigella. The first report of sexually transmitted enteric infection is from this paper in the American Journal of Tropical Medicine and Hygiene. So this was a publication by the professor of parasitology in New York, who was presenting at a conference and talking about how Manhattan in the 1960s was experiencing various epidemics of tropical infections due to mass migration from other parts of the world. Within this publication, he describes this small group, so 4 individuals, which in the diagram are highlighted as F1, F2, R1 and R2. F1 and F2 are 2 men who live together, and R1 and R2 are also 2 men who live together in different parts of the city. And these 2 couples have contact with each other. The language, of course, back in the 1960s around same-sex sexual behaviour is fairly difficult to interpret because there's no explicit sexual history. So, for example, he uses the word acquaintance and roommates, and he talks about shared experiences rather than sexual behaviour, but I think that's what he's talking about. So, as you can see from the diagram, F1 was diagnosed with Giardia, then Entamoeba histolytica as denoted by EH, and various other non-pathogenic parasites, and then again a little bit later was diagnosed again with Giardia, and his roommate or acquaintance also was diagnosed with a similar group of infections, and across the city R1 and R2 also were diagnosed with Entamoeba histolytica, and he surmised from this very early sexual network, only containing 4 individuals, without any real detail of the demographics, that these infections were being sexually transmitted. So this is actually very interesting. Paul Abrahams was another infectious disease doctor working in the US in San Francisco, who described in the Journal of the American Medical Association in 1972 a young man who presented with watery diarrhea and was diagnosed with both Giardia and Entamoeba vermicularis, and he in his paper noted that his exterior countenance suggested of a homosexual preference, and surmised that these 2 infections, so Giardia and Entamoeba vermicularis or threadworms, were acquired sexually. So let's fast forward 10 years, and in the mid-70s we've experienced a bit of sexual revolution. Clinicians are finding it easier to articulate sexual behaviour and sexually transmitted infections amongst men who have sex with men and other same-sex partnerships, and their language is less ambiguous. And here we've got a publication in 1977 by King Holmes, who led a group in Seattle for 30-40 years, and I believe is still working at the date of this presentation, and have done some fantastic work in STIs, particularly around men who have sex with men. And he, in this publication, described a case series of 6 men who all were diagnosed with Giardia lamblia around the same time, so contemporaneously, and were also found to have other parasitic infections which may or may not have been sexually transmitted, and they surmised in this paper that Giardia is sexually transmitted. This is Selma Dritz, who was a public health physician and epidemiologist working in the San Francisco area in the 1960s, 70s and 80s. She was instrumental in describing the sexual transmission of HIV in the San Francisco area in the early 1980s. But before she did this, she wrote some very interesting papers looking at sexually transmitted enteric infections. So she was looking at laboratory confirmed cases of Shigella and hepatitis A, and she noticed that there was a massive disproportionate number of men who were diagnosed with these infections in San Francisco, and with her epidemiological work she was able to show that these men were gay men who were transmitting both Shigella and hepatitis A domestically. In other words, they hadn't imported these infections from other parts of the world. And this was the first reports of Shigella and hepatitis A being sexually transmitted. What's also interesting is that in one of her studies she showed that there was significant antimicrobial resistance in these cases of Shigella, and of course this is an important issue now, and I think this really highlights how easy these organisms, particularly Shigella and the bacterial proctocolitis, can acquire antimicrobial resistance. It's important that we remember that sexually transmitted enteric infections don't occur in the same way that chlamydia, gonorrhea and syphilis do, in an endemic type of way within populations of MSM, and that we often experience outbreaks of these infections which are eventually short-lived usually, and what we don't understand is what happens in between when there are potentially undetected outbreaks because we don't have good surveillance for these infections, or some of these people are asymptomatic with their infection and continue to transmit these organisms. And so I'll talk a little bit about this. What has been fantastic over the past 5-10 years is our ability to use whole genome sequencing to not only map the transmission of some of these infections, particularly Shigella, but also to understand some of the antimicrobial resistance patterns and spread of some of these infections. And this is thanks to work from researchers all over the world who are able to collaborate with each other. And so I'd just like to acknowledge some of these people on this slide. So a really important issue in my view is the asymptomatic transmission of particularly Shigella, but also some of the other bacterial infections. And there's been work from all over the world, particularly Australia, in London and in the Netherlands, looking at asymptomatic sexually transmitted enteric infections, of which there is a significant amount of. So certainly in some of these studies they showed up to 3% of these men had asymptomatic Shigella, and up to 10% of them had some form of enteric infection. So more work is needed here to understand the dynamics of asymptomatic enteric infections. There are some important concepts here to understand around antimicrobial resistance, because certainly Shigella can acquire resistance in two ways: either de novo mutations due to antimicrobial pressure, that's either from advertent or inadvertent prescribing, so people taking antimicrobials from over the counter, but also from environmental antimicrobials such as those used in cattle. But also these organisms can acquire antimicrobial resistance genes from other similar organisms using horizontal transfer. So most of the antimicrobial resistance genes in Shigella are in plasmids, which are circular bodies of DNA occurring in the cytoplasm that are independent of the nucleus, and they reproduce independently too. And these plasmids are small and can actually transmit from one organism to another. They don't have to be the same genus organism, so if we see transmission of plasmids from E. coli to Shigella and vice versa, for example. And this is very important because this process drives rapid horizontal antimicrobial resistance transmission. We do understand now more and more around the genetic codes which confer antimicrobial resistance or are associated with antimicrobial resistance, such as the ones on this slide. So we understand the ones around cotrimoxazole resistance, quinolone or ciprofloxacin resistance, macrolide resistance, particularly azithromycin, and more recently the extended spectrum beta-lactamases or penicillin cephalosporin resistance, of which probably the most important genetic marker is the CTX-M27, and Shigella, which have this particular genetic marker, are resistant to ceftriaxone. So there has been outbreaks of ceftriaxone resistance Shigella over the past 3 or 4 years, including in London. Probably the most important of recent years is this large outbreak, which is really well characterised from Melbourne in Australia. So this is Deborah Williams's group who have published this data of 54 MSM who all had phylogenetically similar organisms, so the same strain, which is at the bottom of the phylogenetic tree. And as you can see from the bars along the side, this is a highly resistant organism, resistant to most of the antibiotics used in this looked at in this study, including having the BLA CTX-M27 gene, which encodes for ceftriaxone resistance. And of course this leads on really nicely and shows that you get international transmission of this infection to this paper recently published in the Lancet. So each bar represents an individual, and it's actually the same strain of Shigella sonnei. The reason for the different colours is that this particular strain has gained this BLA CTX-27 gene, and therefore has become resistant to ceftriaxone. So this particular phenotype is a highly resistant organism. Fortunately, most men or MSM or individuals who acquire Shigella, particularly sexually, do not need antimicrobials. However, as you can see from the right hand graph, which is all the individual cases, some of them did end up in hospital, which is the red box, and some of those people did require antimicrobials because they were septic, and of course clinicians seeing them were very limited by with the antibiotics that they could use. So I hope this has been an interesting short presentation focusing on the history and some of the current issues in sexually transmitted enteric infections. I hope you enjoy the rest of the programme.